gut is divided up into these protozoans according to their motility. Remember when we were talking in that intro deck about protozoans having basically 4 different types of motility. Amoeba that move by extension of pseudopods, kind of the amoeboid type motility. Flagellates that have flagella, move by whipping and or lashing those flagella around. Ciliates, which there's only one we're going to talk about, but they move by beating their many cilia together in unison. And then the last one are the ones that are essentially non-huddled, they have punctoplexa. If they have any motility at all, it's sort of like a little bit of body flexion that gets them where they need to go, which is basically like minute positioning around a host cell. So we'll start first with the amoeba, and a quick review of cysts and trophs is what we're talking about those more. The troph is that actively replicating form, it's a feeding form, it's a metabolically active form. So if you see the troph, that's an indication that there's an active feeding going on, and there's usually or potentially some pathology associated with that. With the cysts, when these intestinal protozoans get ready to transmit from host to host, they will transition from their trophozoite form into that cyst form. So generally, the farther they get down the intestinal tract, they start seeing more and more cysts getting ready to be released from that human host into the environments that can be picked up and transmitted further. With reproduction, usually this is asexual by binary fission. That's all we're going to talk about with the amoeba today. So, they just divide their nuclei, expand their cytoplasm, cytokinesis occurs, they branch off into separate cells. In terms of the clinically important intestinal protozoans, there are really 2 or 3 big ones, but there's a lot of commensals. So this is a list of very common, mostly commensal amoeba that can be found in intestinal tracts, usually on a transient basis. Our immune system tolerates them pretty well because they don't do a lot of damage. What they feed on, these commensals that aren't doing us a lot of damage, are the bacteria in your gut. So they'll eat things like lactobacillus and other bacteria in your gut. There's a lot of species that belong to this genus called Entamoeba. So one of those is Entamoeba coli, E. coli, not to be confused with the bacteria Escherichia coli. Okay. Entamoeba coli, Entamoeba dispar, Mushkowskii, Bangladeshi, that's the E. coli complex. Pilecki, Harmanii, Indolamax, a different genus, Indolamax nana, Iotamoeba bushleae, Chylomastix, Mesnili. So those are all kind of things that are not uncommon to see in stool specimens. Usually they're from asymptomatic patients who are not diarrheal. We've talked about this a little bit, but the O&P exam that we were talking about mostly the last couple lectures with helminthic infections and looking for eggs. Now we're talking about finding cysts and trophs. So that timeline applies where you have that short 30-minute window if you want to find any trophs in this. The slide goes into a little bit more detail about some of these stains too. Trichome strains are used oftentimes when we're looking for intestinal protozoans. That's what we're seeing on the right here, this panel of different images of protozoans. The trichome stain is nice. You get sort of like a magenta stain for the nuclei. And nuclei are important diagnostically, so it's nice to have that feature that's differentiated from that more bluish turquoise counter stain. Chemotoxin stains are also popular. Modified acid-fast stains, like we would use for nocardia, we use those sometimes too. So quite a few images in this short deck have some modified acid-fast stains as well. We have some nice rapid antigen tests for common intestinal protozoans like Giardia and Cryptosporidium. Inexpensive, widely available, pretty good sensitivity most of the time. PCR, that's also in the intro deck, particularly for those major pathogens like Cryptosporidium, Giardia, and Entamoeba histolytica. And then occasionally, you can detect these parasites with endoscopy and biopsy, not very common. We used to do something called an enteroc string test. Anybody heard of that test before? For duodenal parasites, you literally have a patient swallow like a capsule that would come, there was like a gel capsule, so it would encapsulate as soon as it hit your stomach and then go into the duodenum and soak up any parasites that were in there. So while that patient had that capsules soaking up parasites. It would be taped, like that string would be taped to the side of their cheeks so they could walk around for 6 to 8 hours that day with that string there. And then at the end of that period, you draw that string back up to the patient, and it should have, we don't do that much anymore, but it's in the textbook. But I was talking to a PA friend of mine, and he was like, yeah, you'll never do that. So it's no longer in there. But it is a test that used to be more commonly done to look for parasites in the duodenum. I guess these other techniques are probably sufficient to diagnose these infections. A little bit more detail, we see here some distinguishing features from common intestinal protozoans. The main thing that's important to look at is Entamoeba histolytica. This is the most clinically important one and the one that we'll spend most of our time talking about. But these other guys, you can see how the trophozoites might be differentiated from them or might be confused with them as well. With the trophozoites, those tend to be a little bit larger and have more of an amorphous shape, an amoeboid shape, not quite as perfectly spherical as some of these cyst forms are. But with these stains, what we're looking for, things like the nuclei, we're looking for the density of the nuclei, its position within this outer karyosome. The cytoplasm itself, is it coarse, is it fine? Size is very important. We always have our micrometers so we can measure these and see where they fall in relation to other parasites. For the cysts, this is a little bit trickier, but still one of the things we can look for to see if we can distinguish these different types of species just based off these pathologies. These are chromatoidal bars that we can see very clearly with Entamoeba histolytica. That's that condensed dark staining bar right there. It's very long, blunt-ended. Entamoeba hartmannii, very small relative to these others. The number of nuclei is important. You can see how many nuclei. It's kind of hard to see with a stain. A little bit easier if you looked at an iodine wet mouth stain. The number of nuclei in Entamoeba coli. But those are the typical features that we look for when we're diagnosing a known. This slide, there's not, the only reason why I kept it is really for these last two bullets. You can disregard the first two bullets about treatments or study them. I don't care. I won't test you on it though. And this is pretty glib, but it needs to be said that in terms of transmission, the routes of transmission are important to know, but they're all fecal oral, or for the most part, they're fecal oral, some of them are sexual. And kind of as we go through these different parasites, we'll comment on their relative resistance to things like chlorination, which can be important to know. Getting really fine filters so you can filter out these unicellular organisms. And this last one in particular, if you've traveled to developing parts of the world, maybe you've seen these travel advisories on, these recommendations not to drink beverages with ice cubes, because the ice might be a source for parasites themselves. And also the consumption of raw vegetables and fruits. A lot of countries, you may travel to them, and if you're just driving roadside, you'll see subsistence economies, they have big, fruit and vegetable stalls and stands roadside like that. And they look fantastic, they have all their tomatoes up there and their onions and their cucumbers and everything's shiny. But they often wash that produce in ditch water. So it might look clean, but it definitely needs to be rinsed at a minimum to get any potential parasites in that water source off the surface of that produce. So kind of keep that in mind as well. The boiling water guidance is very good. You can kill cysts by boiling water, so that's good guidance. Oftentimes people don't bother to do that. But yeah, that's good guidance in general. Okay, talking about the most clinically relevant member, at least in terms of severity of infection, Entamoeba histolytica. So this is common all over the world. Don't have numbers on how many cases there are. Usually it's fecal oral contamination, ingesting those cysts in contaminated food or water. When you ingest those cysts, as soon as they get triggered by your digestive tract, they lose that cyst wall. They become trophozoites once they hit your small intestine. Trophozoites, what trophozoites do, they start replicating. Most of these species are going to be handled pretty readily by your immune system, so they'll be killed by your immune cells. And many of them that don't get killed will still be non-pathogenic, A, because their numbers are kept pretty low, and B, because they can eat things that won't hurt you, like the bacteria in your gut. It doesn't affect you at all. Where it does affect you, and we don't have a good understanding on what causes some of these Entamoeba histolytica tropes to do this, but they can start eating your intestinal mucosa, and that's when you start getting some of these diarrheal symptoms, which can be potentially pretty severe. So they start eating your intestinal mucosa. If you have chronic infections or heavy infection, this erosion and ulceration of your intestinal mucosa can get to be pretty bad. That's where you start to get bleeding, so you can get amoebic dysentery associated with this organism. And sometimes so bad that you have extraintestinal spread. So hemorrhage, extraintestinal spread, that is unique for Entamoeba histolytica. I can just skip really quick while I'm talking about it onto this next slide that goes into a little bit more in depth about extraintestinal infections. Those tropes, once they've penetrated through the intestinal barrier, they can go any number of places, but where they generally end up going is the liver. And when you have liver infections with these amoeba, those can be really, really bad infections. You can sit there and kind of just chew through liver cells, eating liver cells basically, causing these characteristic flask-shaped ulcers where they, at least in the intestinal, before they penetrate through, and then they start eating the underlying mucosal tissue. So that's like classic for Entamoeba histolytica. Going back to liver damage that they do, it can be some pretty severe abscesses that form in the liver. To diagnosis, you can do aspirates, like liver aspirates, withdraw that tissue, which gets so necrotic that it's described as anchovy paste. It's like this brown necrotic liver paste that comes out of the patient. And in there, you should find lots of trophozoites as a specimen type. You can see some of the damage that's been done to this person: intestinal tissue, liver damage here. So those can be fatal. You can get secondary infections too that can contribute to fatal cases from extraintestinal amoebiasis. Diagnostically, you would have indications of extensive liver damage, elevated liver enzymes, ASTs, ALTs, C-reactive protein levels, jaundice potentially. So going back to just normal road intestinal infections, which most of them, if they're symptomatic at all, it's just mostly going to be intestinal infections. But you get usual symptoms, sometimes pretty severe pain. There should be a comment in there: abdominal pain, diarrhea, some cramping, maybe not blood. And then if you do have liver abscesses or liver involvement, then pain in that upper quadrant, swelling, inflammation, and the immune system trying to clear it out. And then for diagnosis, again, we can see those cysts and tropes that most microscopists are trained pretty well to differentiate cysts and tropes. More importantly, differentiate them from other species. So O&P is really important. Those rapid tests and PCR. And if it is in the liver, we can also do imaging studies as well. Just one more slide on the distinction between histolytica and coli. You don't need to go into any details here, but one of the takeaway points is, while it is possible for an experienced microscopist to differentiate them, it's tricky. The size of the tropes and the cysts, you'll note, has overlap between Amoeba coli and its histolytica. So that can be of limited use. The coarseness of the pattern of peripheral nuclear chromatin, very fine detail there, whether it's fine or coarse or clumped. The karyosome, this is a feature that I find most helpful, and even that can kind of lead you astray, but is it eccentric or is it in the center for histolytica? The most important thing, generally speaking, is if you find, it's like a smoking gun signal, if you find erythrocytes inside of the trophozoites, though. If you do find erythrocytes, that's considered to be feathers out of the cat's mouth type of signal that it's a pathogenic Entamoeba histolytica. So coli does not ingest erythrocytes, but tell what the erythrocytes look like once they're ingested. So tricky stuff. In terms of the transmission pathways for E. hemolytica and all of the other intestinal trophozoites, it's basically a very pattern. There's nothing that is too distinctive. Generally, these leaves get more in the cyst form, but tropes are possible. And what you ingest is almost invariably going to be that cyst form. It needs to be the cyst form, or it will probably get destroyed. What's against your stomach. And one last species. This is still talking about an amoeba. This is Blastocystis hominis. This is one of those that's kind of on the right on the margin of whether or not we consider it to be a pathogen or a commensal organism. It's a disputed reputation because we sometimes find it in asymptomatic people, and then it gets, when there is nothing else we can see in people who are symptomatic, it kind of gets blamed as being the etiology of that. But we don't have, in terms of satisfying Koch's postulates, we don't really have a good connection with Blastocystis. So it's something that we pay attention to if we find it in OMP preps. It's super distinctive. In this trichrome stain, you can kind of see it has a large nucleus, small cytoplasm, and the peripheral nuclei. This is why it's nice to do a trichrome stain. You can see those 4 nuclei very clearly, magenta staining nuclei. It's easy to pick up, but harder to pin down as the etiology if somebody has diarrheal symptoms. Moving on to those flagellates, Giardia duodenalis is the most clinically important, and it's a very clinically important protozoan writ large. It has a very characteristic trophozoite morphology. Sometimes people say it has a smiley face. Sometimes people say it has an old man expression on it because the nuclei on it, the way they're positioned, looks like a face. Its motility, it has all these flagella, these 8 flagella beating around, is distinctive. This is a falling leaf motility. You can see that in this GIF. Kind of just tumbles around like that. So we'll talk about that one first. Dientamoeba fragilis, this is what we were talking about yesterday a little bit when we were talking about pinworm infections. No cyst stage, just trope only. They are difficult to detect, but if you do see them, they have those distinct 2 nuclei, Dientamoeba. That's the name, fragilis, fragile 2 nuclei. And then finally, T. vaginalis has a distinctive morphology as well. We'll take a closer look at that. That also has no cyst stage. So 3 flagellates. Okay, with Giardia, this is a pretty common infection. You'd be surprised if many of you in this room probably had giardiasis and maybe not known about it. It's associated with a couple different risk factors. One of those is being outdoors and camping for extended periods of time, recreationally. Like if you are going out on a week-long backpacking trip or something outside, long enough where you can't bring your own water supplies to carry you through, and you might have to take water supplies or get water from streams and lakes and things like that, particularly streams. So Giardia is common in those streams. There are many animal reservoirs, so it's called beaver fever because beavers are one of many reservoir species that get giardiasis. Muskrats, cats, lots of animals do. But famously, they'll set up a camp and you're downstream of a beaver dam, and there will be Giardia in that water. So there's strong association with beavers, but many animals carry this organism. In the military too, I must say, like, talking about recreational camping, but we don't really do recreational camping. We've all been out there in the field, right, where you are out there for a week or more. Not Air Force.
You're very right.
Okay, well that's my Army students then. When you're out there, not only do you have to contend with getting a water source, which the Army should provide clean water for you with water flows, sanitation teams, treating with iodine tablets, and all the things that should happen, right? Don't always. But then there's the matter of just going to the bathroom all the time. They should also set up field sanitation stations where you have hand washing stations with soap. That always doesn't, that oftentimes does not happen, right? So you have soldiers who are coming out of latrines and, never mind what we were talking about yesterday with hookworms and death. talking about the act of getting fecal contamination on your hands and not being able to wash it off. So you get more bacterial, viral, and parasitic infections in those field conditions. Yeah.
Does it make a difference, nine times out of 10 those hand washing stations
Yeah, so I'm not an expert in that, but non-potable water sources, even if it's gray water, should be, there's like black water, gray water, good water, whatever, different grades. Just because you can't drink it doesn't mean you can't wash your hands in it. It shouldn't be so dirty that your hands are dirtier after you wash your hands in it. But, I don't know, are there any environmental officers or former environmental officers in here? Yeah, okay, what do you think though? Any comment on that? I mean, yeah, don't drink out of the hand washing stations. Correct. Yeah. For what, hand washing?
Yeah, so
Okay, yeah, but gray water, right? Yeah. So yeah, not being able to get your hands clean adequately, and campers are bad at this too, and if you're out in the field, you have your little fighting positions at nighttime, you're pulling guard, are you gonna walk like 500 meters away to go to the positions, or are you just gonna go to some bush nearby you that 300 other soldiers have used in the last, I don't know. I mean, just imagine where you went to basic and AIT. Those training areas are reserved and used constantly, huge turnover. And this is what I was always thinking of, like when I was pulling guard or out in a fighting position, that people are using the same spots to go to the bathroom outside, not using the trains because they are kind of too lazy. And you see like mounds of used toilet paper in these areas. These areas are just like filthy for human waste. And then going back to the recreational campers, for those of you, yeah.
If I may, a quick story.
Yeah.
About something like this. First deployment in Afghanistan, we were doing a clearance off of this a small village. We cleared the village, we kind of take a break with our partner force guys, do the same. There's like this little small creek that runs through the middle of the village. Everyone goes to take a break, one security, whatever. I'm making my rounds. I see 3 different partner force people doing 3 different things in this creek. At the very top of the creek, someone's peeing in it. The guy in the middle is washing his feet, and then the third guy is getting water to make tea. So,
Well, at least no one was defecating in that, sir. I kept waiting for that to happen. But yeah, that's
common, yeah.
classic. All going downstream. Yeah. Good stuff. Good stuff. Just real quick on recreational campers again. Anybody been out to Yosemite before? Like vacation out in Yosemite? No? Well, it's like a zoo out there. You go out there to be in nature, but it's like a small city of campers out there. So lots of humans going on their day-long hikes, and regardless of whether or not they're porta-johns, if there are, that's fine. But if there aren't, people are using the same locations for privacy in the daytime, right? So it's like everyone's using that tree or that rock to go to the bathroom. It just, even in those instances, it gets to be really dirty. And then mountains are disastrous ecologically. People like to go hike on Rainier and Whitney and Shasta, like the big mountains in the US. And that too, I mean, massive diarrhea all over those mountains. Yeah, but I'm serious. Plus, when you're at high altitude, your immune system starts decreasing in function a little bit, and altitude causes altitude sickness. And if you're doing a long hike, like up Kilimanjaro or something like that, it's like a 7-day hike, or the Andes or Himalayas, you are toting your own water supply, so unless you're and filtering and chemically treating your water supply, might be compromised. I'll just say, this is very common. Giardiasis is rampant on some of those mountains. Personal experience. Okay, so enough about the ecology of giardiasis. You get that you can associate that a lot with camping. Also, with just pools, there's a little bit of chlorine resistance as well. Very low chlorine resistance with a lot of these different intestinal protozoans we're talking about. But keep that in mind that this has standard 1 to 2 parts per million chlorination levels. Many water treatment facilities have that. If there's resistance to that, you can get some giardiasis. In terms of the life cycle of these guys, you're ingesting the cysts. The cysts have 4 nuclei in them. And when they inject cysts into those trophozoites, let's say the small intestines, those basically become like 8 different trophozoites. So there's an expansion in the numbers immediately upon excistation in the small intestines. And then once they're trophozoites, they continue to divide. So longitudinally, they split right down the center and divide by binary fission. They get to be thick, especially if you have a compromised immune system and you're not able to keep those numbers down. On the next slide, we'll see how thick they can get. But over time, you can get some pretty significant pathology, pretty serious diarrhea. So the trophozoite form is when they're actively feeding, mostly associated with watery diarrhea. They're not invasive, so you won't see blood. But as they continue down the intestinal tract, so the closer they get to exiting the intestinal tract, the more they tend to form those cysts. So they'll convert from troph to cyst at that point. Asymptomatic infections usually where you have a relatively small dose and you've not repeated exposures. But when you start to get appreciable doses of Giardia in your gut, they can get to be thick. So this is like a blanketed monocanal trophozoites over the intestinal epithelial tissue. When they get to be that thick, your intestines can't function like they should, and you get difficulty absorbing particularly fats and carbohydrates from food. The inability to absorb fats from food results in one of those classic symptoms of giardiasis, which is called steatorrhea. Steatorrhea means fatty stools, basically. So the stools that are released at that point are enriched in fat, they are buoyant, they smell characteristically bad, and they're greasy. There's a distinctive giardiasis type of diarrhea that results from that. Lots of cramping, just a pretty severe type of diarrhea when you have a severe infection like this. In terms of pathogenesis, when those trophs hit the small intestines, they need to be able to adhere very closely to the epithelial cells, so they get washed away immediately through peristalsis. So they have on their anatomy, they have a sucking disc right there, which is basically like coated with carbohydrates and lectins that allow them to form that attachment to those cells. This electron micrograph is also crazy. It's super detailed. You can see how adherent it is to those target cells. So healthy people, typically over time, your immune system, secretory IgA, macrophages, neutrophils will take care of this infection within a week, maybe 2. Chronic disease, you get more serious disease, particularly if you have an IgA deficiency. Intestinal diverticula. This is when parts of your intestines are not folded up properly, so you have a harder time clearing out Giardia from those areas. So that can cause chronic infections as well. Okay. Dianthomema fragilis is also an intestinal pathogen. Still technically considered a flagellate because it has this little undulating membrane on the surface, but it doesn't have the motility that Giardia has. So this is the one that doesn't really have a cyst form, and a lot of our current thinking, we don't have a huge amount of evidence to nail this down, but there seems to be that association that it hitches a ride in that pinworm shell right there. For that reason, it's able to move from host to host and survive. Transmission, fecal-oral route, basically the same transmission as pinworm infections themselves. Fecal-oral route, occasionally oral-anal route, that's a potential route of exposure. A lot of these infections, this is not a super pathogenic organism, so many of these infections are asymptomatic. No tissue invasion, but maybe some minor diarrheal symptoms. The trophs are on the smaller side, 5 to 12 micrograms. Intermittent shedding is difficult to find sometimes. It's fragile and not released in huge numbers, so it's important to concentrate the stool specimens for usually considered to be relatively minor importance. Trichomonas vaginalis is the last of the flagellates that we're covering. This is the one that has those 5 flagella up front and 1 in the back for steering. Very common worldwide. Prevalence studies have put 5 to 20% of women having this particular pathogen, 2 to 10% of men. Probably a lot of underreporting of this underreporting disease, and men tend to be less symptomatic, more asymptomatic, so the numbers are lower there. But because of that, they might be an important reservoir population. It's always important to treat both partners if you detect Trichomonas vaginalis. It has a couple nicknames, T-vag, trick sometimes. It too has a distinctive motility. If you see it when it's still fresh in its trophozoite form on a slide, it's very lively and moves a lot. So it is transmitted through sexual intercourse. It is capable of, it likes to adhere to and attach to the epithelial cells of the reproductive tract. In women, it could be cells in the vaginal tract or cervicitis in cervix and cause cervicitis. In men, it can attach to epithelial cells in the urethra and occasionally the prostate as well. So symptoms associated with symptomatic infections of T-vag in women are a little bit different, mostly related to discharge, itching, dysuria, painful burning sensation during urination. Occasionally transmission can be vertical, so congenital transmission. And if babies are born with Trichomonas vaginalis infections, that can be associated with things like pneumonia and conjunctivitis, or low birth weight in some cases. If men are symptomatic, they can get symptoms like urethritis, like a burning sensation during urination as well, but usually not as serious one. Did you have a question?
Ma'am, without, like, actually doing lab testing, would it appear different at all from what the
Oh, that's a good question. So in women in particular, the only thing that comes to mind is perhaps that discharge, which in this case is described as green and frothy. Maybe I've never heard that discharge referred to as green or frothy. So it might not be super helpful. I'd definitely order a lab test to confirm that diagnosis. I wouldn't base it off of pure opinions, but the discharge is a little bit different, and that is probably due to the characteristic destruction of those epithelial cells in this, as well as some immunopathology. This is a target. Trichomonas vaginalis is a target on the biofire estimator panel, so it's up there with all the heavy-hitting bacteria and viruses or STIs. You can see those tropes on smears. The reason why it doesn't have a cyst stage is because it is transmitted in such a intimate manner, sexual transmission. There's no survival time needed outside the host. There is, up there you can see it says some fomites, so there might be a limited period of time where it survives on things like towels, and you can transmit it that way, but it's a very short window. I'm talking like 30 minutes or less. So primarily, by far, the primary mode of transmission is sexual intercourse. And more pictures of what you can see on a stain specimen. You can see that axing down the center of these tropes. And you can even see some of the details of the flagella as well. It's kind of a pretty organism. Okay, we are on the ciliates now. This is the only ciliate protozoan that we're covering today. This is Neobalantidium coli. It used to be Balantidium coli. It's not a strict human pathogen. It is carried by pigs and other animals. Pigs are, particularly in underdeveloped parts of the world where pigs and humans cohabitate closely. You get sometimes outbreaks of this particular organism, Balantidiasis. Fecal-oral transmission, sort of the same process once it ingests cysts and they exist in the small intestines. But because it's a ciliated organism, its pathology is slightly different. Nothing too sophisticated, but that trophozoite, once it develops in the small intestine, it has it's called cytostome, and that's basically like a little bit of an opening where it can consume nutrients. Those nutrients are usually like intestinal villi. So it can actually, it's not eating solely bacteria, and it does cause some damage, some direct tissue damage. Plus the cilia allow it to kind of burrow into the intestinal epithelial cells a little bit, so there's some damage that way as well. But yeah, it's usually fairly limited. Sometimes that damage is sufficient enough to cause a little bit of bleeding. So you can have a little bit of blood in stools, maybe it's apparent, maybe it's occult. You could use that occult blood test if you wanted to help further your diagnosis. But the cells released from this infection are super, super distinctive. These guys right here, these trophozoites, I'm sorry, if you do see trophozoites, these look like right here, the kind of oblong shaped trophozoites. The cysts are also fairly distinctive in that they're huge. Both of these forms are huge. So 200 micrometers among the easiest protozoans to identify through microscopy. And it's easy to see cilia on the surface of them as well. Okay, we're actually almost done with this deck. I've got 10:40. Why don't we take a 10-minute break now, and then we'll cover the last remaining intestinal protozoans.
Okay, Tom. We're gonna wait for you to keep going until Which way do you go?
Which would you lean? And then I thought you were anti-mouthwash because it washed away all the minutes. Pro from insult.
Alright guys. Talking about this last category of motility, the last three organisms that we're talking about are all intestinal protozoans, and they are all sporozoans, which are a type of protozoan P. complexa, which captures some more clinically relevant members. We'll talk about this next week. So when we talk about Plasmodium, the causative agent of malaria, Toxoplasma, these two are all sporozoans. So very similar type of motility, or lack of motility, more accurately. They have a complex life cycle that toggles back and forth between sexual and asexual reproduction. It's confusing, so I'm kind of glad we're dipping our toes into it today, talking about these intestinal sporozoans, because when we start talking about malaria next week, it will hopefully make a little bit more sense. You won't have to know the finer details of reproduction, but just as an overview, like how this happens, would be important. And the fact that for protozoans, the fact that they can reproduce sexually is important as well. This cartoon here shows cyst with sporozoans inside, sporozoites. This cyst behaves like a cyst, protects them and allows them to be transmitted host to host. And then if you look at these sporozoites themselves, look at the shape of them, they have an apical complex here, where my laser pointer is on those organisms. That apical complex is the part that will eventually interface with the target cell, and through some mechanism allow them to invade into that target cell. So they have like an orientation and a polarity to the sporozoites. These sporozoites are also haploid. What does that mean in terms of the number of chromosomes that they have? Yeah, they've got one set of chromosomes. So when they undergo this asexual reproduction, which is the most common form, it's called schizogony. It means that they're basically just reproducing that one haploid genome, and so the offspring also have one genome copy. So the offspring also haploid. So these guys, for example, if they're haploid, they will undergo schizogony and they will produce many, many other copies of them. And that sporocyst will grow with a number of different organisms that allows them to amplify the patterns. And then occasionally, you'll have these specialized parasites that'll be called, they're gametes essentially, they will further develop into either male or female. If they're male, they're called microgametes, and if they're female, they're called macrogametes. The males will fertilize the females, and you will have production of a zygote that is temporarily diploid, like two copies of the chromosome. And then it undergoes a reductive form of division called meiosis, and this progeny are back to being haploid again. So that's kind of that cycle between sexual and asexual reproduction that sporozoans can go through. We'll talk about that a little bit more in detail, but after they undergo sexual reproduction, the resulting offspring is this oocyst form, and this oocyst is the form that allows it to be transmitted from host to host, resistant, environmentally stable form of oocyst. And inside of it has those maturing sporozoites that are infected. The three members of these intestinal sporozoites that we're covering today, they all start with C, so try not to get them confused. There's ways to distinguish them, but it's easy to get them confused as you're studying for this exam. Okay, in terms of morphology, a couple images that help you distinguish what they look like under different types of stain. At the top here is Cystoisospora belli, Cryptosporidium, and Cephalospora chiatinensis. Top, these are wet mounts, so these aren't even stained with iodine. So just look at the size difference. Cystoisospora is quite large, usually 20 to 30 micrometers, and it's ellipsoid. The shape of it is very distinctive relative to the other protozoans there. Cryptosporidium is the smallest one. It's a fractal, teeny tiny, 46 micrometer parasite, and cisform, and then Cephalospora chiatinensis is sort of about half a medium. It's around 8 to 10 microns. You do stains like a safranin stain, you can see how that stains the nuclei very well for Cystoisospora belli on the left. And with these two organisms, this is a different stain. This is a acid-fast stain. We'll talk about some problems that we sometimes encounter when we do acid-fast stains for those organisms. So first we'll talk about Cystoisospora belli, which is probably the least clinically important of those three. It's also the hardest to describe. It doesn't really have a lot of good descriptions to sink your teeth into. Worldwide distribution, increasing frequency in both healthy and immunocompromised patients. And the reason why we bother to say that is because we're probably getting a little bit better at picking up this disease, and that's probably because of the fact that it's opportunistic in AIDS patients. So the disease that it causes, the diarrhea that it causes, is particularly severe in AIDS patients, but that's true of the rest of these sporozoans as well. In terms of this infective cycle, you ingest this mature oocyst, fecal oral transmission. Could be food, could be water. You ingest it. Once it hits your small intestines, these sporozoites, which have matured inside of this oocyst, can burst out and start dividing through schizodism, that asexual form of reproduction in the intestinal mucosa. That division contributes to some of the pathology and the damage to the intestinal epithelial layer. Some of those will go on to become gametes and undergo sexual reproduction, so you'll get fertilized oocysts resulting from that process, and that is ultimately what you shed in your feces as the infective or transmissible part of this organism. It's the oocyst. That oocyst is not immediately infected. It has to mature a little bit longer, but this maturation period, once it's left closed, is pretty short. Short enough for some important transmissions between patients. So in order for it to be infected, it does have to be have to have some sporocysts inside of it. But this can happen in a window of a couple hours, maybe a day. Most of the transmission is ingestion of fecal oral water, but if you see your oral anal sexual contact is another transmission form. And the implications there are, like, if you particularly if you have poor hygiene and this is not like these organisms aren't being washed away through showering, you transmit it over the course, like a delayed course of 6 to 12 hours. Okay. So this does happen. It's not nearly as frequent as ingestion of food or water, but it's something to kind of be aware of. In terms of diagnosis, I had mentioned before, you get, let me switch to this other slide that talks about it, that shows these images a little better. You do get this giant ellipsoidal sporocyst. So those are fairly distinctive. This is from the CDC's website. They have a really good division of parasitic diseases if you ever want to peruse their website. But they do make some mistakes, and they've mislabeled this. This is not a safranin stain. This is just normal iodine stain. It's a wet mount. But regardless, you can still see it. It's very hard, so it would be beneficial to stain it. You can see that giant ellipsoidal sporocyst there. Here too, I doubt that this might be safranin, but it's definitely counterstained with blue stain. So you can see that single sporoblast in the center, and then a trichrome stain here. And lastly, by virtue of the fact that these parasites can autofluoresce, you can see them under UV light. Here you can see distinctive sporoblasts inside a single cyst. So this would be an infected form right here. Size, shape, whether or not there are like 1 or 2 sporoblasts, all of that would be of interest for diagnostics. The next one is Cryptosporidium species. The 2 primary ones that we are concerned about are Cryptosporidium hominis and Cryptosporidium parvum. Parvum, this one is carried by different animals, particularly livestock, but other animals as well. So you can be interacting closely in rural areas. If they get cryptosporidiosis, there's going to be associated more with parvum. Hominis is more of a human-specific parasite. So the transmission is fecal-oral route, person-to-person. If we look at some of these risk categories, this too is somewhat resistant to chlorination. There have been outbreaks linked to swimming in chlorinated water, where kids have diarrhea, Cryptosporidium. I should stop picking on kids. It's not like adults can't go swimming with diarrhea as well. But people who have diarrhea are swimming, and a bunch of people get sick as a result. So, high risk groups, people's animal handlers, particularly with parvum, children, men who have sex with men, MSM. This is a pathogen that can be readily infective. So if you look at this, here, if you look at this life cycle here, notice that what leaves the body, this mature oocyst with a bunch of sporozoites inside of it, that's the same form that infects you. So you can have auto-infections, and for that reason, it is easy to transmit sexually depending on different sexual practices. So keep that in mind. Immediately infected auto-infections are possible. The most common transmission source is ingestion of contaminated water, though. I mentioned before that massive outbreak in Milwaukee in 1993. I looked up the numbers freshly, and that is one of the biggest wastewater outbreaks in history. 400,000 people got cryptosporidiosis because of that filtration in the city's municipal water supply. Milwaukee is not that big of a town, it's like 1.2 million. So a lot of people got sick. And at that time, in 1993, when that outbreak happened in early spring, like February, March timeframe, or the AIDS epidemic was still such catastrophe. So the people, many people did not know they were HIV positive until they contracted cryptosporidiosis. One of those opportunistic pathogens, which is particularly severe in immunocompromised patients. So a lot of them found out that they were HIV positive during that outbreak. And of the people who were progressed to AIDS, as defined by their CD4 counts and opportunistic AIDS-defining illnesses, 200 of those people who had AIDS got really serious infections, and 69 of those people died. So 35% CFR because of that one outbreak. It cost the city of Milwaukee millions of dollars, lots of medical bills, just a disaster. That kind of underscores the immunocompromised population, but going back to healthy people and outbreaks with healthy people, waterborne outbreaks, we have been doing some surveillance with the British military in Kenya, up in Kenya. They go there for a lot of their training rotations. So they send cohorts of their soldiers, about like 1,200 person cohorts, and they rotate there for 2 months or so. We do diarrheal surveillance in their populations, so collaborate with their RDCM. They had a massive outbreak of cryptosporidium in this British military population because they were swimming outside, because they were ingesting water that was contaminated with it. They had something like 170 or 180 troops get cryptosporidiosis. And it's not like a life-threatening disease if you're immunocompetent, but it's still a big deal for military welfare as well. The British leadership is much more permissive of their troops, so they would let them do a lot of things that our leadership probably doing with legs. They have pubs and bars on their FOBs, and it's just wild. But big outbreak. With most of those healthy cases of cryptosporidiosis, some of them will be asymptomatic, some of them will be mild, but they are self-limiting, so you don't need to treat them necessarily. It will go away within a couple days. Self-limiting diarrhea, no blood, no mucus, no phage is intestinal epithelial cells. But the problem again is when you have immunocompromised people who cannot clear this infection, and they have incredibly severe diarrhea, on the order of like as bad as we're talking about with polyposis, where you are losing 5 to 10 liters of fluid a day. You simply cannot survive that on a long-term basis. Chronic infection, Cryptosporidium. So people do die from dehydration and from electrolyte imbalances. 50 plus stools a day is just unsustainable like that. So it can be serious. For diagnostics, this is a common target on PCRs. For O&P preps, this is that small one. It's easy to miss sometimes because it is so tiny, 46 milliliters. Also, never mind what's saying about the reproduction. We already talked about that. But on the note of biopsy here, if you were doing a modified acid-fast stain on Cryptosporidium, it's a good stain to use, but there is variable staining. Some of these oocysts do not take up that stain very well, so you get variable staining patterns. Some of them don't stain, so it's easier to miss. Like some of these, you can see that magenta stain, and that can be a sign that the infection is resolving when you get these ghost oocytes that don't stain at all. They're just not viable anymore. So that can be a good sign as well, but it's tricky for diagnosis. This here is kind of interesting too. If you look at the blue arrow that I'm highlighting there, that's a yeast cell. So it's not that much bigger than yeast cells. On the right, red arrows are Cryptosporidium, and on the left is blue arrows, yeast cells. Okay, and the very last one is Cyclospora chiatinensis. Chiatinensis is probably the most clinically relevant species in this TST, and there are other species as well. Chiatinensis is mostly human specific, so the other species can affect animals and contribute to reservoir populations and its persistence in the human population. But like I said, chiatinensis is the most widespread one. The nice thing about chiatinensis, Cyclospora chiatinensis, is that it does have an easy couple of associations to make with it. It is, in contrast to Cryptosporidium, which is more associated with contaminated water consumption, with Cyclospora, most of the big outbreaks that we get related to this parasite have to do with produce that's been contaminated. So it's like almost every summer, about big outbreaks where hundreds of people are sick, and it's usually linked back to something like cilantro or basil or leafy vegetables, raspberries, something like that, a common source, oftentimes imported from other countries, sometimes from the US, that causes these multi-state diarrheal outbreaks to occur. So kind of keep your antenna pointed and oriented towards that as a common source, not that other things can't transmit Cyclospora chiatinensis, but oftentimes implicated with contaminated fruits and vegetables. And note also that what leaves the body is this immature oocyst, unsporulated. So it requires further development before it is infectious. That means you cannot auto-infect, no sexual transmission. For symptoms, nothing particularly distinctive about the symptoms, just generic diarrheal symptoms, healthy people, self-limited, although it can be prolonged. It might take your immune system a while to get rid of this infection. And with immunocompromised, the diarrhea can be very, very severe. I don't know if it's life-threatening necessarily, but it's not considered to be one of those defining illnesses like these other pathogens that we've talked about. It's just worse in those patients. Thoroughly washing vegetables is an important pro tip. With leafy vegetables, that's really hard to do because their surface area is much greater, but you can at least try. In terms of microscopy, this tube has variable staining, modified acid-fast stain. Characteristic are these oocytes that sometimes pick up stain and sometimes don't. It's called mottled. You've heard of mottled flesh. There's a lot to look to it. We use that same adjective to describe these mottled pink oocysts that sometimes stain this way. Otherwise, they're fairly hard to ID. They're in that intermediate size range, 8 to 10 micrometers, refractile and unstained wet mount, like these three animals right now. Refractile meaning shiny, and then that model and stain acid fasting. Okay, any questions on the testing sporozoans? Okay, I've got some questions for you. Just to kind of help you, I think those are difficult sometimes, but these are classic vignettes. The answers are on your slides, but do connect the salient features in these clinical vignettes to how you would perhaps get a hint as to what the etiology of this is. So read through anything is important for this vignette. It might take you off to some particular etiology. This size, super small size. Okay, anything else? Oh, yeah, non-bloody diarrhea, so not invasive, frequent water diarrhea. Anything else? Say. Yes, an animal exposure to possible zoonosis. Rural farming community. She's heading soon, perhaps with livestock. Child. Okay, so if we put all those teeny tiny hints together, and she puts as a daycare as well. If you put those tiny hints together, what do you think that sounds like of those three organisms I just talked about? Yes, parvum. That was the only zoonotic disease, and it also matches his size. That's probably the strongest clue here. The size of his oocysts. Okay. This vignette, we've got a 5-year-old male with history of AIDS, CD4 count 75. He has persistent chronic diarrhea for over a month. So he's got more severe symptoms of weight loss, fatigue, and cramping. We've ruled out bacterial etiologies. We have numerous large 20 to 30 micrometer acid fast oocysts in his stool. They're ellipsoidal in shape, and they autoexpress after you subject them to UV. It's like every hint under the sun. What do you think? Belly, yeah, cystoisospora belly, the one that is kind of the hardest one to describe, and everything we could pull for that. This is also the one that is considered to be the least clinically relevant. It's the fewest number of cases. And then lastly, we've got a female, 20-year-old female with travel history to Guatemala, notable consumption of fresh salads and fruit. She has normal diarrheal symptoms, cramping, diarrhea, fatigue, nausea. Medium-sized oocysts, variable staining, fasting, and some of those will fluoresce, autofluoresce if you like. What do you think? Cyclospora caritanesis. Yep. Okay, so those types of vignettes, not those exact ones, but those are some things that might help you prepare for intestinal sporozoans. Any questions? Yeah.
Let me see what I can do on my
Okay. I do have an answer, but I really hope it wasn't offend anyone. Okay, so yesterday he asked me if I could go by a country, what would I go to? But he didn't qualify it with like.
avoid.
Oh,
In the
avoid?
realm of like, in the realm of like infectious disease, microbiology,
Oh.
which country would you avoid at all?
You can answer that one.
Oh man, you guys spent
Yeah,
6
right.
hours thinking where to go. Now I need another 24 hours.
Well, where would you go?
Thank you. I'll answer that one. And I'm gonna work on these. I would go to Brazil. Yeah.
You would go.
I would go to Brazil. So I've spent a lot of time. I've lived in Asia, lived in Africa, lived in Europe, lived in North America most of my life. I spent very little time in South America.